All right, so muscles that allow you to increase the volume of your of your chest cavity. The diaphragm is the main one. That's you know, the majority, especially in quiet breathing, the diaphragm is the main muscle that is helping you change the volume of your thoracic cavity, which then, because of that pleural section, increases the volume of the lungs, which creates slow pressure in the lungs, which is what actually drives the movement of air into your lungs during inspiration. The diaphragm, what nerve innervates the diaphragm?
Phrenic.
The phrenic nerve, which comes from which plexus?
Cervical.
The cervical plexus, exactly. Um, right, and for this, the pons and the medulla are generating the basic rhythm, up in your brainstem. It's different than the heart, right? Your heart keeps beating because it's got its own internal pacemaker, that SA node in the wall of the right atrium. But the lungs actually have the circuitry up in your brainstem, just sending a message down your phrenic nerve to activate the diaphragm so you keep breathing. What are other muscles that you can bring in to help change the volume of your chest cavity?
Intercostals.
Intercostals, and you have external and internal to either expand or compress. What are some other ones?
Serratus anterior.
Serratus anterior, which can pull on your ribs. What other ones?
Scalenes.
Scalenes pull up on your ribs as well. What's another little one from the coracoid to your ribs?
Minor.
Latissimus dorsi.
Minor. So there's a variety of muscles that do this. Okie dokie. And then it says, name 2 distinct ways your respiratory system can deal with the dust you breathe in. So we talked about a number of things going on here.
The cilia with the sticky mucus.
Exactly. We have that respiratory epithelium. It's got sticky mucus, which can trap the dust like a fly in a fly paper or something. And then the cilia that sweep it to the back of your throat so you can swallow it, get rid of it. What are some other ways we have to deal with dust you're breathing in?
Macrophages.
Yeah, the alveolar macrophages, the dust cells, which are down in the lung tissue itself and clean out the alveoli if any little bits of soot or something get down in there. What other things do we have?
Mucus.
So somebody said the mucus already, that mucus that was on that respiratory epithelium.
The hairs in your nose to trap the dust as it comes in.
Yeah, what do we call, what's the fancy word for nose hairs?
Vibrissae.
The vibrissae. The vibrissae is the fancy name. And then we also talked about, you've got these explosive air movements you can use if you need to clear out your lower or upper respiratory passages. What are those called? An explosive blast of air coming out through your nose.
Sneezing.
Sneeze. You can sneeze, or a cough is an explosive blast of air coming out your mouth to clear out your lower respiratory passages. So you've got all sorts of tricks up your sleeve to try to keep your airways open and keep dust out and all that. Next we says, why are surfactant cells important in your lungs?
They help keep the alveolar sacs open by diffusing the water tension.
Yeah, so they are the things that break up the water tension, so it makes it easier to inflate the alveoli. If you have water tension, it pulls in and makes it hard to inflate. Excuse me. We talked about, part of the problem with the COVID disease is surfactant cells get hit, and without making surfactant, it's harder to inflate your lungs. The more and more they look at this coronavirus, the more and more it's attacking all sorts of other things in your body. It's pretty nasty. I highly recommend not getting it. It seems to be making people more likely to get blood clots and strokes and ruin their heart muscle, get heart attacks and all sorts of stuff. So it's not just about like your lungs, it's intestinal stuff, neural stuff. Like one of the new symptoms that's on the books as a typical symptom is like, anosmia, loss of smell, which is definitely getting more into neurological stuff, which is kind of weird. Anyway, back. What kind of tissue lines the nasal cavities and respiratory passages? Yes, that everybody should remember this from the very beginning of the semester, the ciliated pseudostratified columnar epithelium with goblet cells. Right, so that's the one that's got the goblet cells to make the mucus. There are also mucus glands underneath that are bringing mucus up as well. And then the cilia sweeping it to the back of the throat, either down from the nasal cavities or up from the trachea and lower respiratory passages. So, any questions about the old warm-up? All right, so our warm-up is done. Other kind of nuts and boltsy things I'm looking here. The next quiz is going to be exactly a week from today. And it's going to cover, the new stuff since this last exam. It's going to cover the endocrine system and the respiratory system and now the digestive system that we're doing this week. So that will be on Tuesday, 1 week from today, on what do we call it? On the 5th of May. So putting that out there. And we can stop sharing this now. All right. So I think now it's time to bite into the digestive system.
Oh, Lord.
What's that? Do you have a question, Victoria? Oh, man, I think she's been, all right. So functions, what are functions of the digestive system?
Break down food.
Break down food. So that is a big part of it, is breakdown, of food. And food, what the heck is food? Food in general is other organisms, whether plants or animals. It could be plants, it could be a wheat plant or maybe an apple tree or who knows what. It could be an animal, maybe a cow that you're making a hamburger out of, or chicken, getting some Kentucky Fried Chicken or something. So, but in general, food are the bodies of other organisms, either plants or animals. And they have all of these important molecules that you need either for structure or for energy, but they're assembled in the form of that particular plant or animal. So the first step of your digestive system is basically to disassemble and dismantle that creature into the basic building block molecules. So breakdown of food, into I'm going to say building block molecules. So individual amino acids, individual monosaccharides, individual fatty acids, and things like that. So that's a big part of our digestive system, is taking in our food and disassembling it into these molecules that we can then use. And then we have to actually bring them into our body. So then we have to absorb those nutrients. So we're going to talk about that. And that can be different in different types of nutrients. Like fats, you have to deal with differently than with sugars, because fats don't dissolve in water. So they don't get absorbed in the same way. They can't be handled the same way. So we'll be looking at the breakdown as well as the absorption. And we'll look specifically at a few different kinds of nutrients. There's a variety of nutrients, but people talk about carbohydrates. Then there's proteins, there's fats. These are nutrients that we talk about as energy producing. Like, these are all things that you can either use as building blocks to make parts of yourself, or you can break down actually to release energy to drive the different processes in your body. So these all can supply energy. But you also take in proteins, so you can dismantle them into amino acids just as building blocks to make your own proteins, right? So, you know, it's a kind of a weird thing when you think about what life is in a lot of ways. It's basically creatures consuming other creatures, digesting them, dismantling them into individual molecules again, then using their genetic code and their DNA to reassemble all of those amino acids and molecules into their own image and their own form, and into the parts and pieces that they need for their body, until something comes along and eats them, and then dismantles their body into individual molecules, which then get reassembled into this new thing. Or even after you're dead and you're lying there, there's going to be little bacteria and fungi that come and dismantle you into individual building block molecules to make their little fungal and bacterial bodies. So it's kind of this fascinating thing when you think about life. It's just everything just consuming itself and repatterning those building blocks, and then getting consumed and getting repatterned into something new. It's kind of beautiful and horrifying to think about. I mean, there are other things we consider nutrients. These would be things that we consider nutrients. There are also minerals. Minerals just mean like elements, like calcium and sodium, and, you know, things like that. Vitamins. Vitamins are actually organic molecules that you need to get from your diet. Organic meaning that they are carbon containing. You know, for this class, you don't need to go deep into what are vitamins and minerals. I'm kind of putting this in here just to kind of flesh out what we mean by nutrients. But in physiology, you need to know what a vitamin is. In anatomy, you don't really need to know what a vitamin is. But it's basically things that help run the metabolic processes in your body that have to be acquired through your diet. They're what we call essential. Water is actually considered a nutrient. It's something you need to absorb through your diet, and we'll talk about where that gets absorbed. So this is the basic layout of the digestive system. And let's start with breakdown. Actually, let's start with the layout. Let's start with the layout first. This person's got really short legs. So the basic thing that's kind of strange about the digestive system is there is a tube that's just running all the way through you. It starts in your oral cavity, goes to your esophagus, there's your stomach, your intestines, your small intestine, your large intestine, your rectum, your anus. It's like poop. So basically, when you eat food, it's never actually in your body. It's always in this tube running through you. People talk about a human being topologically a donut. You're basically a donut, like a hole at the top, a hole at the bottom running through you. So this tube, we have two different names for it. It can be called the gastrointestinal tract. AKA the GI tract, AKA the alimentary canal. We're gonna look at this tube in a lot of detail. We're gonna look at the basic structure of it, and then we're gonna look at how it's specialized in the different regions. You know, it looks different in the oral cavity and the esophagus compared to the stomach, compared to the different sections of the small intestine, compared to the large intestine. So we are going to look at how it's all similar, but then we'll also look at the specializations that make it different along the length. And we'll also talk about the different processes that are happening along the way. Then there are also going to be accessory structures. And this will be like your teeth. It's going to be like different kinds of glands. You're going to have salivary glands, and you're going to have the pancreas and the liver. So we're going to talk about those as well. We'll talk about the GI tract. We'll talk about these accessory structures. And we'll talk about both the breakdown and the absorption that is happening along the way as we do this whole thing. So let's see, before we get into the details, let us go into the more generic structure of the GI tract. So let us do that. So this basic layout is going to be the same everywhere, but there'll obviously be modifications depending where you're at. There's going to be a lumen. Lumen just means the internal part, the hole of the tube. And then the lining of it is always going to be some kind of an epithelium. You know, exactly what that epithelium is going to be different in different places. For right now, in fact, for most of the digestive tract, it's simple columnar, but it doesn't matter. We're going to see it's not necessarily. So here's my epithelium. Just underneath the epithelium is loose connective tissue called the lamina propria. And I should mention this epithelium, I've drawn it here as a nice smooth circle, but it's often heavily folded into all sorts of little folds and fingers or pockets or grooves or holes. We'll look at all the different versions of heavily folding to increase surface area as we continue on. And then underneath the lamina propria is there's actually going to be 2 layers of muscles. I'm just going to make it as a single thing for right now. It's what we're going to call the muscularis mucosa. I'm going to write scant smooth muscle. This muscularis mucosa is not a major player in moving things around. It's more about maybe puckering the surface a bit or something. But it's not, we're going to see a much more beefy muscularis that is going to be involved with squishing food around and moving it from one part of the tube to the other. So all of these together, this is called the mucosa. The mucosa is an epithelium, which is the thing directly lining the lumen. This lamina propria, which is loose connective tissue just under the epithelium. And then the muscularis mucosa, kind of a thin layer of smooth muscle that can let it like wrinkle up a bit or stuff like that. Below that is, in fact, what's going to be just underneath the mucosa?
Submucosa.
Exactly, the submucosa. In the submucosa, this is going to be dense CT. And here you find all sorts of stuff. There's going to be glands, there's going to be blood vessels and nerves and who knows what. So submucosa, dense CT with all sorts of stuff in it. Next comes the muscularis externa. The muscularis externa is the muscle that actually does squish your food around and move it around the tubes. So the muscularis externa has 2 layers. It has one layer of smooth muscle fibers that go around in a circle around the tube. So I'm going to kind of draw. So what I'm drawing now is called the muscularis externa. Actually, let me. The muscularis externa, and it has 2 layers. There's a circular layer. And then there's a longitudinal layer. Which, if in a cross section, is just going to look like looking at a bunch of these little, right? If along the long way, so these are going back. So this longitudinal layer, if you're looking at the cross section, it just looks like a bunch of kind of dots because it's smooth muscle cells that have been cut off in cross section. But if you're looking at them along sideways, then they have, right, this would be a cross section, but it's actually a smooth muscle cell that's going this way. So that's the muscularis externa. It's got a circular layer, which is the ones that go and can tighten up, constrict the tube. The longitudinal layer of the smooth muscle can kind of pooch out the tube. So muscularis externa, very different from the muscularis mucosa. Muscularis mucosa is just a little bit of muscle right underneath the mucosa. Muscularis externa is kind of the workhorse of smooshing things around the tube. And then what covers the outermost side of everything on this? The outside of what envelops all your digestive viscera? Keeps it from rubbing against itself.
Serosa.
The serosa, exactly. The serous membrane. So that's going to be the last part. Serosa, which is just basically simple squamous epithelium with some loose CT underneath. And we talked a little bit before about how when it wraps around, you create these double layers called mesenteries and all that. We'll talk about that in more detail. There are a few places where the tube is not covered in the serosa, not covered in the peritoneum. The esophagus is not in peritoneum. It's actually just going down through your mediastinum. There are parts of the beginning of the duodenum is actually pooching out behind the peritoneum. So any place where the tube is not coated in the serous membrane, then it's got just dense connective tissue on the outside. And I don't know if, do people remember the generic term? We used it when we talked about the outside of blood vessels. What was that dense connective tissue on the outside of blood vessels called?
Adventitia.
Exactly. So like the adventitia, that's only going to be on very specific places like the esophagus or the duodenum, we'll see. But in general, most of the GI tract is surrounded in serous membrane, so it can slip and slide. All right, so this is the basic picture of the GI tract. We're going to see some areas have different kinds of epithelium. Some it's thrown into finger-like folds. Sometimes it's thrown down into pit-like invaginations. There's different kinds of glands you find in the submucosa, but it's usually looks similar to this. Two more things I want to add in here. I'm going to introduce it on another page and then we'll come back here. So the enteric nervous system. So sometimes people call this like the primitive gut brain. Whoop, I didn't mean to do that. So the idea here is for some of the most basic things that your processes in your digestive tract, your brain is not involved. Remember, we always talked about, in basic reflexes, if we think about what is a reflex, there's some kind of stimulus, there's some kind of integration, figure out what do we do with that stimulus, and then there's some kind of response. And we've always been thinking about the CNS as being the thing that integrates, that figures out what is the response going to be. But it turns out in the digestive tract, you have a couple of neural networks that also do integration and will take care of basic digestive processes without using the brain. So let me introduce them to you. There is a plexus of neurons in the submucosa here. That's called the submucosal plexus of Meissner. And there's another plexus in between the circular and longitudinal layers called the myenteric plexus of Auerbach. So these are nerve plexuses. And I've kind of run out of room to name them here, so I'm going to put them back on that other sheet. So just from their name, you can figure out which one's which. Submucosal plexus is the one in the submucosa. Myenteric is the one in the muscularis externa, in between the two layers of smooth muscle. What kind of stimuli is this enteric nervous system going to be able to detect? What kind of information do you need to run your digestive processes?
Chemoreceptors.
It's going to be chemoreceptors. We're going to have information about the substrates of digestion. So we know what kind of enzymes do we need to break it down. We'll have chemoreceptors for the products of digestion, so we can know that we're done breaking down and it's time for the next step. We're going to have chemoreceptors for pH, because a lot of these enzymes in the digestive tract, function optimally at a particular pH. What other kind of receptors are we going to need to run digestion?
Mechanoreceptors.
Yeah, mechanoreceptors. Stretch receptors. I know that it's time to do something because my stomach just got all stretched out because there's food in there now. Or there is still something stuck in my throat and I need to keep moving it down, because I can feel the stretch receptors are telling me that the tube is stretched out. So you have chemoreceptors, mechanoreceptors. The enteric nervous system can do this integration, and the response is going to be either some kind of muscle contraction or, glandular secretion. If the integration is totally within the enteric nervous system, then we say it's a short reflex. So if there's a reflex where the stimulus is in the digestive tract, the integration is in the digestive tract, and the response is in the digestive tract, that's called a short reflex. It is possible to have reflexes that do involve your brain. And then we call it a long reflex. Again, we don't get into that so much in this class, but even just thinking about food will actually start activating secretion in your stomach and stuff. So your brain definitely has, a role to play in your digestive processes. It's just that it doesn't have to. There are reflexes that are wholly within the walls of the digestive tract.
A question for the long reflex, would it ever be integrated in the spinal cord, or does it always go up to the brain?
I don't know for sure there. The ones I know involve the brain, but that doesn't mean that there aren't some in the spinal cord. So,
Okay, thank you.
So glands, we're saying the response can be gland secreting. That's pretty straightforward. A gland secretes saliva is pouring into your mouth, or pancreatic enzymes are pouring into your intestine. For the muscle contraction, I want to look at, there's two basic types of muscle contraction that you get in the walls of this GI tract. So again, remembering that muscularis externa is going to be our major player to do this moving things around. And there are fibers that are circular that can squeeze the tube. And there are longitudinal fibers going along the long way that can kind of pooch out the tube, expand the tube. And the two main kinds of coordinated contraction of the smooth muscle are called segmentation and peristalsis. So let's look at that. So we'll start with peristalsis because that one is, everybody knows this one. Peristalsis, for propulsion. So when you want to move something along a tube, for instance, in your esophagus is going to be pretty much pure peristalsis. Your esophagus is a tube going from your throat down to your stomach. And let's say there is some bolus of food there and you need to keep moving it down. Basically, what's going to happen is you'll squeeze the muscle right above it, the circular layer, which is going to then push it. You can pooch out the part right below it, and it's going to basically push it a little further down the line. And then if we look in the miracle of time-lapse photography here, we can see now it's a little further down. Now you contract the smooth muscle in the circular layer right above it where it is now. And you do the longitudinal layer, pooch out the layer below, the little plate below. It's a little further down. And so on. So, this is why you can be just lying on a couch horizontally and someone feeding you grapes. And you swallow the grapes and the grapes still get to your stomach. They don't need gravity to get to your stomach. They're getting squished along by peristalsis. You could swallow standing on your head and this food would still get to your stomach because it's just getting squished along. Um, you know, it takes a little while for your food to get into your stomach after you swallow. Takes like maybe 5 to 7 seconds. It's like kind of whoop, 2 and 3 and 4 and 5 and plop. Now it's in your stomach. When you're swallowing, what happens if this thing gets stuck here and the wave continues on? You know, the wave continues on, but the food is still like stuck up here. Probably you've felt that before. It's like, ooh, there's still something in here. It didn't make it all down through. Fortunately, it's going to initiate a secondary wave of peristalsis that's going to come down and catch it and then hopefully continue carrying it down. I'm sure, has anybody not ever experienced that where you swallow something and you realize it didn't go all the way down, it's still stuck somehow? Nobody's ever experienced that?
I've had that before.
Yeah, so it's not uncommon. So peristalsis for propulsion. The other thing is segmentation. This is for mixing. You know, this is more going to happen like in your intestines, where you've got your food. Oh, let me make it blue again. And it's getting mushed up with all these digestive juices. And here the idea is not about moving it along. It's about mixing it and smushing it so the food has a chance to break down. So here we're going to like contract where it's kind of pooched. We'll pooch where it's contracted. And the food's kind of getting smushed. And then we do it again. We pooch where it's contracted and contract where it's pooched. And whoop. And pretty soon it's just a big mess. So think about smush, 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 smush. Think of like a washing machine or something. You're basically squishing it back and forth and mixing it up, allowing the digestive enzymes a chance to do their work and break apart the food into the building block molecules. You know, and in reality, it's rare you're doing purely one or the other. You know, your esophagus really is purely peristalsis. But for the most part, you know, once the food gets down into your digestive tract, you're smushing it along to mix it, but then also moving it along, a little propulsion to keep it moving to the next stages. So, you know, sometimes you get like a mix of both of these. But it's worth knowing the two main types of contraction and what they're used for. Let's see. Oh, hold on one sec. I realize I don't have my clock going, so I don't even know what time it is. Oh, we're doing good. All right, so now I think we've got our ducks in a row to actually start the digestive journey. Okay. Oh, we need teeth. Okay, this guy looks a little goofy. So obviously, and actually I usually have food. Hold on a second. All right. Here is some food, some little toasty thing. So obviously it's going to start with me putting it in my mouth and chewing it. All right, first step, chewing it up. That is a type of breakdown. Am I actually breaking it down into fundamental building block molecules when I'm just doing the chewing?
Don't you have enzymes to break down starches in your spit?
Right, so the chewing itself is not going to break down into actual molecules. It's what we call mechanical breakdown. So when we talk about breakdown, there are two different types. There's mechanical. That is what I'm doing with my teeth. I'm taking this larger physical thing, breaking it down into smaller physical things, increasing surface area, but it's not gonna be what's actually gonna get me my building block molecules. So this is like chewing. This is also like in your stomach. We're gonna talk about the stomach as basically smushing the food back and forth, kind of kneading. Think about like if you're kneading bread dough in your stomach. So this could be like kneading in the stomach. You know, when you first chew down, you swallow your food, it's not instantly turned into molecules. If you've ever like swallowed, you know, ate a big plate of spaghetti and then barfed and realized, oh my God, I can see all my chewed up spaghetti lying there on the ground, right? If you vomit pretty quickly, not that long after you've eaten the meal, you can recognize the chewed up food that you swallowed. It's not like, you know, so once the food lands in your stomach, it takes it a while. It's going to get smushed into a paste by the muscles in the stomach, just smushing it and smushing it and smushing it and smushing it. So mechanical breakdown is this kind of either chewing or kneading, but it's not taking you down to individual molecules yet. The other kind of breakdown is the chemical breakdown. And this is using enzymes. So this is how you get the dismantling of the food into individual amino acids and things like that. So chemical breakdown is also, so, and the thing Casey mentioned saliva. Saliva is going to play a role. So you're going to have some chemical breakdown starting in your mouth already. So let's talk about salivary glands and saliva. And teeth as well. Yeah, I'll do the salivary glands and then we'll do the teeth. So you've got a couple of salivary glands. You've got salivary glands that are basically on your cheeks that kind of feed in behind your molars here. These are called the parotid glands. They're kind of like right here. You have a sublingual gland underneath your tongue. Which basically pours in right under your tongue. And then there's a submandibular gland, which pours in kind of also kind of on the front of your tongue. And submandibular, sublingual, and parotid salivary glands all make saliva, although slightly different versions. So let's talk a bit about saliva. One is mucus. And then the other part is, so basically amylase. From serous cells. Amylase, breaks down starch. So when you think about what the saliva is doing, one thing is it's just kind of like this stuff to help bind your food into a slippery ball so you can swallow it. Like, ready? If I take this cracker thing, no, it's dust, right? You can't swallow that, you choke. So if that, you chew it, you got the saliva coming in, binds the food, what we call, into a bolus, bolus meaning little ball. Yeah. Right? I got a little ball of food now, a little slippery ball. I can swallow that. That'll go down my esophagus and it'll be fine. Right? If I tried to swallow the cracker dust, I'd be in trouble. So part of what the saliva is doing is binding the food into a bolus so you can actually swallow it. But then the other thing that the saliva is doing is it has this amylase, so it's actually starting the chemical breakdown of the food in your mouth. So it doesn't finish the job. The amylase in your saliva gets deactivated the moment it hits your stomach. You're going to have other amylases. There's a pancreatic amylase coming in to the small intestine that is going to continue the job. But the very beginning happens in your mouth. You can actually, if you start chewing starchy foods and leave it in your mouth long enough, you'll start tasting it sweeter because it's going to start dismantling the starch into simpler sugars in your very mouth. Under the microscope, salivary glands are really cool. I like them a lot. They have mucus cells that look a lot like goblet cells. The cells, the serous cells that make the amylase, so mucus cells look kind of like mucusy. Serous cells are very filled with granules. They're very dark because they're making their little enzyme. When we get to the lab part, I'll talk more about what salivary glands look like. Parotid glands are mainly serous cells. Making basically a watery enzyme solution. Sublingual are mainly mucus. And these submandibular are more mixed. me see if I can do this. Give me a second here. Yeah, actually, I'm going to, so here, this is kind of giving you a sense. Here's like, these are the parotid glands. I kind of removed, I removed a lot of his teeth and stuff, left his gums in place so you can kind of see. You can see the parotid glands are basically here in the cheeks. There's this thing, it's called the parotid duct. The parotid duct is going to then carry the saliva and dump it in right behind your upper molars there. So those are your parotid salivary glands. Underneath the tongue here, there's the sublingual glands. And you can see the sublingual gland basically pouring in under the tongue. And here's the submandibular gland, which is basically underneath your mandible here. And it has a little duct which is pouring in kind of to the front of your tongue here. So this kind of gives you a sense of what the salivary glands are like. And again, teeth. The teeth are the other thing that we are going to look at in a little more detail now, and as well as the gum, this gum, the gingiva, right? Gingivitis, most of you probably heard of. So we're going to talk about the teeth and what kind of teeth, what teeth are, how many you got, and what holds them inside your jaw. Teeth. So teeth are, when you are born, obviously you usually don't have teeth. You get what's called primary dentition. These are your baby teeth. And then, as sure you all realized at some point when you were kids, you used to have your secondary teeth start coming in, the secondary dentition. These are going to be your permanent teeth, you know, and there are little organs called enamel organs that make the teeth. You know, after you make your first teeth, there's still the enamel organs. So you got your second round and you make your secondary dentition. And there's going to be 32 in all. Primary dentition is only like 24 teeth. You've probably, you know, when you see a nature show about sharks, they just keep making new teeth. You know, they don't worry. Our enamel organs go away after the secondary dentition. So if you lose teeth after your secondary dentition is out, you're just gonna have to get a fake tooth. Other rodents, like if you're a rodent, your front teeth never stop growing. They just keep growing and growing and growing and growing. That's why they have to gnaw all the time. If they don't gnaw, their teeth will actually go through their jaw and make it so they can't eat. So you have to allow rodents to gnaw or else it's deadly for them because their teeth just never stop growing. Just so you don't think that there's different, you know, that all teeth are the same. Very briefly, you know, if we like look at the mouth as kind of four quadrants, upper and lower, right and left, then the teeth are all kind of mirrored. You've got your incisors, which are basically like for kind of slicing through things. your canines or cuspids. Which are more like for ripping. Your bicuspids or premolars. And then you have three sets of, or three true molars here. So this is 1, 2, 3, 4, 5, 6, 7, 8. 8 times 4 is going to give you 32 teeth in all. These back molars erupt the last if they erupt at all, and these are your wisdom teeth here. And if you look at, it says I don't have the cat skeleton here, but if you look at different animals, their teeth are optimized for what they eat. If you look at a cat's teeth are basically all these the sharp little ripping teeth. They don't have any of these like grinding teeth like molars. If you look at like a moose, all it has are incisors and molars. It doesn't have any of these canines because it's just kind of slicing vegetation and grinding it down, right? So we have teeth that, and actually a whole digestive system that's optimized to be very omnivorous. We can eat almost anything and manage it. And we'll talk later. The one thing we're not very good at extracting nutrients just from pure grass and stuff like that. No mammals are. For those animals like cows and deer and things that live off of grazing, they usually have symbiotic bacteria that will work with them to help extract more nutrients from the cellulose. But we'll talk about that later. So teeth, primary dentition, your baby teeth. Sometimes they call them the milk teeth. Your permanent or secondary dentition. Let's look at a tooth. A tooth is actually a lot like a bone. And the dentine, the thing that is actually the structure of the tooth, is a lot like bone tissue. So let's look at what a tooth actually looks like. Tooth, up close and personal. Okay, it's Tommy the tooth. So inside the tooth is what's called the pulp cavity. And there's going to be blood vessels and nerves and different kind of connective tissue that's in there. It's kind of hollow and it's got little pores coming out the bottom, so the blood and nerves can connect up into there. Its bottom part's called the root. The top part is called the crown. Most of the tooth is made of this stuff called dentin. Dentin is very bone-like. It's like bone, Um, instead of having osteons, instead of having those like Haversian systems, it has these little, they're called dentinal tubules. And those are so the little cells that are making and maintaining the bone, the dentin can kind of reach in and work in there. So when you look at the tooth, it actually has this kind of cool stripy thing that you can see. Dentin. The crown, the top of the crown is covered in enamel. Enamel is the hardest material in the human body. Assuming that you have fluoride in your diet, it will use fluoride. I don't know if you remember, hydroxyapatite was the mineral that makes up your bone. Enamel becomes what's called fluoroapatite. Which is even harder than bone. So this makes the crown, the outside crown of your tooth, very abrasion resistant. So it doesn't like grind down as you're eating stuff. So that is Mr. Tooth. And now what we're going to do is how the tooth gets anchored into the jaw. So up close and personal.
Does the enamel regrow, or like can it regenerate itself?
Say it again.
Can the enamel regenerate itself?
I'm not sure. I think up to a point the tooth has some ability to work on itself and maintain itself, but I don't know how far that ability goes. So I don't know. Let's look at the tooth in the gum. So first the bone. And let's say this is your mandible or your maxilla, either one, right? It's the bone. There's some socket for this thing. There's Tommy the tooth. And there's a variety of things. There's this, in fact, you don't need to, you're not responsible for it, but there's actually this coating that is going to anchor these little ligaments called cementum along here. And then there are little ligaments that actually anchor from the bone to the tooth. They're called the periodontal ligaments. There's different regions of them, but for this, you just need to know they're periodontal ligaments. So this is going to be a synarthrotic fibrous joint, right? Synarthrotic because it's not going to move. Fibrous because it's little fibers that are connecting from between the tooth bones. What's the official word for this special joint?
Omphosis.
That's the gomphosis. And then you seal it with soft tissue. This is the gingiva, which in common tongue is just your gums. And that seals this little gomphosis up here. Some of the things that go wrong, we can talk briefly about issues. Gingivitis, inflammation of the gingiva. When that happens, you can start getting kind of that retraction of the gums start disconnecting from the tooth. making it more vulnerable for things to get down in there. If it gets bad enough, you get what's called periodontal disease, where there's bacteria that are actually in here and you can actually lose your teeth. So gingivitis is inflammation of the gum. You start getting recession of the gum. And if you lose this good seal, you can actually get infections down into the actual joint that are gonna like make you lose your teeth. Other things, dental, does anyone know the official word for cavities?
Tonium.
They're called dental caries. Dental caries is the official word for cavities. And what they basically happen from is if there's some food someplace, usually it's more in a crevasse. Let's make it in a crevasse. And then bacteria start metabolizing that, and they have acids that they produce as byproducts. And then those acids can start dissolving the tooth. And that's what a cavity is. One of the kind of ironic things is, sugar isn't such a bad thing for getting cavities. It'll make you fat for sure, but sugar is water soluble. So sugar, if you have sugar on your tooth, it usually washes away, so it doesn't stick around for the bacteria to eat. It's carbohydrates, like this little piece of toast is way worse for me if I don't brush my teeth well, because the starch is not very water soluble. So the starch just kind of sticks in the little crack there, doesn't dissolve in my saliva, and is a thing that the little bacteria just can keep munching on, make acids and dissolve my tooth. So saliva is really important to keep you from getting cavities. The saliva washes away bits of food and stuff, and it also is antimicrobial. Your tears, I talked about tears having lysozyme, that antimicrobial material or secretion that kills bacteria. Your saliva also has that. So the saliva, in addition to washing away stuff, it's also antibacterial. I was talking to a friend of mine who does, he works with people who have drug abuse problems. And I was asking him about like the classic image of, the kind of people addicted to meth and losing all their teeth and stuff. And he was saying that it's really true. And he's saying part of what's going on is, sympathetic. A lot of those drugs are sympathetomimetic. They activate your sympathetic nervous system. And part of sympathetic is suppressing your digestive processes. So you're not salivating as much. And when you're not salivating as much, you're not having all that antimicrobial and cleaning saliva irrigating around, and you're more likely to get cavities, in addition to just not brushing your teeth and having poor hygiene and stuff. But, so it was kind of interesting to kind of put together some of the anatomy, physiology with some of those, tales of, kind of the speed freaks losing their teeth kind of thing. It was kind of interesting. So where we are. So what's that, Mr. Tooth? Oh, he's saying it's 2 p.m. So I think that means we probably have time for our break. So let's take a break. Are there any questions? One of the things I mentioned a while ago that I was going to finally do is more formally lay out the points for the final grade. So I finally did that. So if we go, let me kind of show you how this works. This way you can get a better sense of how everything is going to be calculated. Assignments. So the basic idea here is now the homework assignments, which includes, that system overview you did in the very beginning, the endocrine overview that is currently going on, the muscle drawing packet, which I actually do have, and I just haven't finished grading, and those different Pearson ones. Those are going to count not exact. They're going to count 6% of your total grade. So the actual points don't matter so much. It's like basically however percentage you got of this goes to become 6% of the total. All the warm-ups become 4% of the total. And I added a rule here, like drop the lowest 2 scores for that. The quizzes are going to be 10% of your total grade. Lecture exams are going to be 40% of the total, and lab practicals are 40% of the total. So that is how everything, and that's pretty much what things were when we first started the semester. I just finally kind of figured out how to formally make Canvas do this. So just kind of putting that out there.
So is that our grades will reflect now?
It should be, yeah, if you look in Canvas, I think this will be reflected in how you see things now. You should let me know. I mean, I can get like a student view, but not a student view of a real student. I have a student view of like a fake student that hasn't done any kind of work yet.
On
Okay.
Canvas, if you click on grades, it has like all your grades like kind of going down the center, and then towards the right, it'll show like the weight of everything.
Okay, so that's good.
Yeah, it's updated, yeah.
The lecture and lab practicals, and we're going to go over the lecture exam just in a few seconds, but just kind of reiterating what I said at the beginning of the semester. Your scores on those, if they're solid, then that's awesome. If they're not, and you're worried that they're pulling down your overall grade, they are adjusted based on your final exam score. The final exam is going to be comprehensive. It'll focus, I like to say, it's about 2/3 will be of the new stuff. But then there will also be, going back to what tissue is this, or what bone is this, or what articulates here, or what muscle is this, or what, going back, or cardiovascular, or looking at the nervous system, all of that. So it's a comprehensive final. And that means that if I feel that you know the stuff by the end, like you have done a solid job on the final, even though you really choked on one of the other exams, what I care about is that you're solid, as we wrap everything up. So I do make adjustments. So if you have a low score in any of the major exams and you do well on the final, then it actually pulls it up. So just kind of putting that out there. Whatever your current score is on things isn't necessarily true if you do well on the final. So I'll put that out there. So let's take a look at Anatomy Lecture Exam 2. This is the one you just took. And we got like the cool cat taking an exam on the computer. It's like your life these days, like everything, you're just like sitting in front of a screen. And we can look at quiz statistics. And what we see You know, high score, 103%. We actually had a few people getting over 100%. Low score though was down 65. That's not good. In general, people rocked on this thing. You know, most people got A's and B's, you know, one down here in the 70% and one below. So if you're down in this region, you definitely want to think about your approach. You know, I've kind of the same spiel I've given before is doing what you've always been doing usually leads to the same results. Or just like unless you know for sure you just haven't had time to study, usually just, well, I'll study harder is not the best thing. It's usually like, how can I study a little differently? If you're up here, then it's all good. If you want to talk to me, if anybody has any concerns or wants, like, you know, talk about approaches to the final, just let me know. I'm happy to Zoom talk with you. All right, so we can just look. I mean, one of the things that's I really don't like about this is that you can't actually look at your exam and see what you got wrong personally, but we'll kind of go through all of this. And if at any point you want to look at your personal exam and see what you got right or wrong, then, you know, we'll schedule like either an office hours or another time. We can do that together. It's just, we obviously can't do that all together on Zoom in front of everybody here. All right, so you have taken the food into your mouth. You are masticating, chewing it up, mixing it up with the saliva, creating this slippery little bolus that you're going to swallow. And so the next step is to swallow, and deglutition is the fancy word for swallowing. And it's going to be voluntary to start with. You're going to use your tongue to kind of push the bolus to the back of your throat. And you have these muscles that we did not have to learn explicitly, but as a group, they're called pharyngeal constrictors. You've got a whole bunch of muscles that can squeeze down in the bottom of your throat there. And they're going to keep squeezing this thing down. So that first part is voluntary. But then once it hits the esophagus, then the automatic reflex takes over. Then it's going to be the enteric nervous system doing peristalsis. So this first part is voluntary. But then once it's going, like I said, it just gets this wave, peristaltic wave carries it all the way down and it plops in your stomach. What the heck is this? Whoop. Okay, so then let's talk about the different sections of the tube briefly. We're gonna do a little, I should have, this is gonna be on TV. Let's see. So this initial part here, the oral cavity and the esophagus, what kind of epithelium do you think is going to be lining those?
Stratified squamous epithelium.
Exactly, exactly. So both of these are going to be places where the food is really rough. It hasn't really been processed, and where it can, you're chewing like some scratchy toast and or Captain Crunch, and both of these are going to be having stratified squamous epithelium for their mucosa. Oops. So when you're looking at the esophagus, one of the things you're going to notice is that the mucosa is stratified squamous epithelium. How about everything else? What is the mucosa? Say it again.
Simple columnar.
Uh-huh. Exactly. So the stomach, the small intestine, the large intestine, they all have simple columnar epithelium lining the lumen. However, there are some differences. Stomach, you have these things, they're called gastric pits. Going down into these glands. So it's not, if you look at the lumen, it's not just a flat line. Like if I think of simple columnar, it's not just like that. Here we have the simple columnar going down, creating these pits where there's glands making gastric juices coming up. When we get to the small intestine, we have what are called villi. Villus is like a little finger. The simple columnar epithelium is actually thrown up into all of these little fingers. When we get to the colon or the large intestine, we have what are called crypts, which are basically kind of like a little, like a gopher hole. Similar to the gastric pits, but not quite. So just to kind of put this out there, because we're going to get introduced to trying to recognize the different layers of the alimentary canal right now. Next class, we're going to look at all of these in more detail. But for today, I just kind of want you to wrap your head around the idea that, yes, we're going to have all those layers. We're going to have the mucosa, the submucosa, the muscularis externa, and then the serosa. But it'll look different in different areas of the digestive tract. If you're up in the esophagus, you're going to have stratified squamous epithelium and not much of this folding or pits or villi. Once we get to the stomach, you're going to get all sorts of these pits with all sorts of weird glands down in there. Once you get into the small intestine, it's like a shag rug, basically. All of these little fingers, and you don't necessarily even, a lot of times they get cut at an angle, so they don't even, it's hard to even tell their fingers. The colon, you have these like gopher holes, basically. I can, yeah, right. If I have a bunch of gopher holes, and I cut it just right, it'll look like this. But the fact is, a lot of times it gets cut at a different angle, in which case the intestine, instead of looking like a bunch of gopher holes, looks like a bunch of flowers. There's the lumen and there's the cells making up the wall. Right, so,
I have a question.
uh-huh.
So in the large intestines, those crypts, does it have glands inside it, or is it just like that?
They have, these are like goblet cells here.
Oh,
And so there's mucus coming up here. And basically it's making a nice slippery, slidey place for your feces to be moving along towards the final exit.
thank you.
So, but both of these pictures, this picture and that picture, both are really typical slides of the colon, especially the mucosa of the colon. So just to kind of put it out there, because they have such weird folding and fingers and pits and crypts, that it can be a little confusing when you first start to look at it.
I have one more question.
Sure.
So this columnars, are they like ciliated or is it villi?
So most of it depends where you're looking. So that's actually a good question here. When we're in the small intestine here, where we want, this is going to be the main place where we're absorbing the nutrients. It's the main place of absorption in the digestive system. And so when we look at, I'm going to take this villus, and look at it in higher magnification right now. So columnar, simple columnar epithelium. Here's the lumen. Inside here is going to be the capillaries. Actually going to be a little lacteal, little lymph vessel. And these typically have microvilli. But which you don't actually see as individual villi because they're too small. So instead they just look kind of like this indistinct fuzziness. So sometimes they call it the brush border. And again, these are not about moving or anything. These are just about increasing surface area. And they don't look like actual little hairs. They're too small. You could only see the hair-like quality if you were looking at them with an electron microscope, but you can tell there's something there, that the surface is not just clean. So we will come back, but microvilli are individual, like if I had a cell, microvilli are the individual little hairs on a cell. Villi are these big kind of fingers made out of lots of columnar cells that, yeah, again, oh my, we're not going to be looking at an actual, if we were, if you are in the lab and looking at an actual intestine and you open it up, it looks kind of like velvet inside. It's actually, it doesn't look smooth. It looks kind of like soft and velvety, because the reality is it's just all these little microvilli. Not villi, that all these little villi increasing the surface area. So when you look at it, even from a macroscopic gross kind of level, you can tell that it looks kind of like velvet or something like that. It's got like kind of a a texture. All right, so I'm gonna do a now. Go back to here. So part of what I'm going to kind of guide you through some of this, but then I think it probably makes sense to just do it on your own. Under this, the histology resource I have at the bottom where it says virtual histology lab, they have some pretty nice slides of the digestive tract. And again, the thing I like about these is it's more of a real experience of starting at low magnification and zooming in. And so you can see the larger overall structure, but then you can also see the details of it. So if you go into the slide box and into the gastrointestinal tract, you can actually see stuff like, esophagus. So here from kind of this big picture view that we can see here, there's a few things that you want to keep track of. Wait, let me find my little, there it is, annotator. Where'd it go? I apologize. Oh, there it is. Okay, drawer. So up here, this is going to be the mucosa. And if I go below it, this is going to be the submucosa. It's going to be dense connective tissue with glands and blood vessels and stuff. If I go lower, this is going to be the muscularis externa. And we can see there are two distinct layers. There's this layer here, this internal circular layer where the fibers are going in along the direction of this thing. And then this longitudinal layer where you can see, you actually don't see nice, you can imagine the smooth muscle cell looks kind of like that. So on this upper part, you're seeing them just going along their length. But on this lower part, you're seeing a cross section. So you're looking at them all head on. This is going to be really common across all the different sections that you look at in the GI tract, in the alimentary canal. It's going to be one layer where they're going in one direction, one layer where they're perpendicular. So this is like a nice introduction to those three layers. And then if we zoom in.
Are they always in that orientation, like the circular on top of the longitudinal?
Circular is always, is the first one. So I can tell you for sure that this is a cross section of the esophagus because these are going this way. If I had a longitudinal section, then the circular ones would look head on. Yeah, so that's a good question. The layer closer to the lumen is always circular. Oh, let me turn off this little do for a second. So then if I zoom in, that's why I love this thing. It's like, oh my God, there it is. There's stratified squamous epithelium. So there's that basement membrane there, the cells. This is exactly what we were hoping it would be. Then you look at here, getting into the submucosa, and it's like, sure as heck fire, looks like dense irregular connective tissue. It's kind of like the dermis of your skin. And there's some arteries in there, classic arteries with smooth muscle and some blood cells in there. This one's probably more like a vein because it's got thinner walls. There's a little arteriole there. Oh, these things, cuboidal walls, these got to be ducts. In fact, these are some kind of mucus gland here because these are what mucus cells, these are like goblet cells making some kind of mucus. Here are cuboidal cells making ducts. So there's glands down here, there's blood vessels, whatever. We continue on. Now we are in the muscularis externa. This is a circular layer where we're seeing the smooth muscle cells that are going along their axis. And then when we get down here, we're now looking at the longitudinal layer that's been cut head on. So you're looking at each of these individual cells head on now. So it looks more like kind of this pointillism thing. And again, there's also connective tissue and blood vessels and stuff that are going through here because it needs, you know, obviously. And then we come out to the outside. You know, this is some loose connective. This is actually in the esophagus. What you're going to basically have is adventitia because the esophagus does not have a serosa on it. So, yeah. I would say, I know this has to be some dense connective tissue, so I'm going to say it is. You know, and again, as we get in here, you could just see like itsy bitsy little blood vessels. They're adorbs, right? So now, in fact, this is probably a capillary right here with one little blood vessel, one little blood cell inside this tiny little thing made out of a little wrapped around endothelial cell. So here's another blood vessel that's been cut more longitudinally, and you can, yeah, it's fun. The more you've learned about what to recognize, the more fun it is to just look around and see all this stuff going on. Gastroesophageal junction. Here is where the stomach and the esophagus meet. Obviously, there's going to be this transition from stratified squamous mucosa, which up here we can see is pretty distorted, but it's the stratified squamous, till all of a sudden it's like, oh my God, there's something super different going on here. This is now simple columnar epithelium with some kind of connective tissue underneath. So we are now officially in the stomach. We have made the transition. So this is kind of cool. Here you can again see the mucosa has just this simple columnar epithelium. Although here, this is kind of what I'm talking about. These are gastric pits that have been sliced at a slight off angle. So if you're looking at a gastric pit perfectly from the side, it would be like, oh, it's going down and there's a pit. But if you've cut it from like a cross angle, you know, it looks like a bunch of little holes. Let me, like here. Like right here, think about this. Here's columnar epithelium coming down into a pit. But what you're looking at over here, where we were a few seconds ago, these are a whole bunch of those pits, but now that have been cut at an angle, like I was showing you on that picture a little earlier. So it can get really confusing looking at these slides. You have to remember that you're looking at a very convoluted surface that is not going to behave nicely with the section. So you'll have some parts where you say, oh yeah, I see the epithelium coming down in a pit. And other times you're just down in the connective tissue looking at just the part of the gopher hole that gets cross-sectioned by the slide. Oh my God, what is this? What does that look like? Does that thing look familiar?
I got gland.
No. Does that look familiar? I know you were looking at things that looked similar to this in the histology.
Is it a nerve?
No, it's got a really distinctive look. And it's right underneath the mucosa. Huh. What things are these kind of dense staining, like follicular structures right underneath the mucosa?
Is that like the nerves and stuff?
No, it's not nerves. Back in the 50s, you go on a date, you like should put your straw in, you go to the shop where you get like the soda. Oh, remember malt? Remember mucosa associated lymphatic tissue? I was like trying to like remember old malt shops or something. This is just a freaking lymph nodule. It's a lymphoid follicle, lymphoid nodule. This is just malt. Remember, you know, we talked about there's formal ones that are your tonsils, and there's the famous Peyer's patches, but they're scattered all over the place. And here we got lucky to just see there's a lymphoid nodule underneath the mucosa doing its job. The stomach has, yeah, don't worry about it now. The stomach is got a special third layer in its muscularis externa. It actually has an oblique layer, which we'll talk about later. So for right now, don't worry about the muscularis externa looking like it might be a little different than the others. But basically, it's the same basic idea here. Mucosa, but really convoluted because you got all those pits and gastric glands. Then you got the submucosa, dense connective. You got muscularis externa with different orientations. And then this is probably just a loose CT going into, it's probably a serosa in here. It's hard to even see. Yeah, I don't want to definitively say that this is, but this might even be the serosa. I don't know. Stomach. Again, here's more stomach. Again, as you can see, depending where you look, sometimes you can see, oh yeah, simple columnar epithelium with these pits coming down in these regions of all these glands. However, if you cut at a slightly different place, you know, it looks like this because now you're actually cross-sectioning the pits. So just kind of putting that out there. When you look at this, the pits are going to look funny. And when we get to the small intestine with the villi, instead of cross-sectioning the pits, you're going to cross-section the little fingers. So anyway, we'll come back. And again, for right now, I don't care that you know the details. We're going to talk about the details of these later. What I want you to be able to do is like realize, oh, this must be the mucosa, but it's really convoluted. That's why it looks so weird. Submucosa, the dense connective tissue, muscularis externa with the circular and longitudinal layer, and in the stomach, there's another layer. So that's kind of what I want you to think about. When we get, the duodenum gets crazy with, let's look at the jejunum. Here, well, this is stained really trippy. This is kind of going back to what Sakai was talking about, the brush border. You can see the cells have this kind of fuzziness on the edge of the columnar cells. That's the brush border there. See that fuzziness? Here, this is a finger, a villus that's been cut. So you're looking at a cross section of a finger. Here's a finger that's more longitudinal section, so you can see the whole finger, right? So here's a finger, a villus, but here's one that's been cross-sectioned this way, so it looks like it's just a circle floating in space. And then here, you can see the submucosa, which is stained in blue, actually gets thrown up into these big, they're called plicae circularis, to give you even more surface area. We're going to look at all the different levels of folding in here, but basically you've got individual cells have folds in the microvilli to increase surface area. Then the mucosa has the villi, so each of these columnar epitheliums are going up and down in all of these little fingers. And then the submucosa goes up in these big circular folds, and the mucosa runs up and down along those. So by the time we get all this folding going on, you've got a surface area about the size of a football field inside of your small intestine. So you have a really intense ability to absorb nutrients from your digestive tract into your bloodstream. Oh, and I should go back. Here is the circular layer. So now we know we have a longitudinal section through the, this going back to Sari's comment. Since this is circular layer, but it's cross section, you're looking at the cells head on. I know that I'm looking at this intestine along the long ways of it, and that the longitudinal cells are looking kind of long and slinky because these are going along the long direction of the tube. So this way I know that I have a longitudinal section of the small intestine here. And these things here, like in between, these are what let me double check. These are very likely parts of the myenteric plexus. Like when you see cells that are stuck in between the circular and longitudinal layers of the muscularis externa, they're usually the neurons and such that are part of the enteric nervous system, the myenteric plexus of Auerbach. We can look, let's look at the ileum. So like the ileum is something you saw when we were looking at the Peyer's patches. There's no Peyer's patches on this particular section, but you can see the same thing. Muscularis externa, longitudinal layer going the long ways, circular layer that's been cross-sectioned head on, submucosa, the dense irregular connective tissue underneath the mucosa here, creating one of these plicae circularis, and then the mucosa on top with the simple columnar epithelium going up and down in the villi. This is all the lamina propria is the connective tissue inside the villus. And inside the villus is where you're going to find the capillaries to absorb your nutrients. It's where you're going to find the lacteals to absorb the fats. And again, if we zoom in more and more and more and more, all of a sudden you see like, oh my God, there's the brush border again. The actual like edges of these columnar cells are kind of fuzzy looking, out of focus, because it's the microvilli for that extra layer of increasing the surface area. Colon. Here we have these crypts. Basically, the columnar epithelium goes down into these long crypts that have lots of these mucus-producing goblet cells. If you cut it at more of an angle, let me see if they got it here. Yeah, like over, I'm going to go to, like, you can see over here. Here's the same little crypts, but now you can see the hole, the gopher hole, and the cells surrounding it, right? So, and you also see a whole lot of lymphatic tissue. This is all lymphatic tissue, more lymph nodules here in the submucosa, more malt. And then if we look here, there's circular layer of smooth muscle and longitudinal layer of smooth muscle. So let me think if there's, so what I would like you to do for today is get comfortable with those different layers. Don't worry. Eventually, you're going to be able to recognize where you are. But for right now, I just want you to recognize which layer you're in. I want you to know mucosa. And in fact, let me see if I can find a muscularis mucosa for you. Hold on. Give me 1 second here. Okay, there it is. If you're looking right underneath the mucosa, it's like, oh my God, there's some little, those are classic looking smooth muscle cells, those little slinky guys here. So this is muscularis mucosa. You know, so right under the mucosa, there is this very thin layer of muscle. Just kind of serves more to kind of maybe wrinkle things, wave the little villi around a little, but not really major squishing of anything. These muscles here, the muscularis externa, are the ones that do the peristalsis and the segmentation. All right, let's see what else we can find. We can go into the slide box and go to exocrine glands. And here's some salivary glands. If we go into the parotid gland, we look around and we see all of these cells that stain with the dark little granules. Those are the serous cells. Those are the cells that make the amylase. Cells that make mucus stain, they look more like goblet cells, and we're not really seeing them so much. And then we're seeing a whole bunch of ducts, little tubes made out of simple cuboidal epithelium. If we move around a little more, you know, larger ducts made out of stratified cuboidal epithelium or stratified columnar. This has got to be a vein because it's got super thin walls and it's filled with blood vessels, blood cells in there. So again, ducts, you can recognize ducts because they're cuboidal epithelium making a tube. You know you're looking at a blood vessel if it's got smooth muscle making up the wall. Again, here's a perfect looking duct, whereas this is going to be probably a vein right here. And again, in this parotid, it's pretty much pure serous. Up here, these are what the mucus glands look like. The mucus cells look like this. They look kind of these gloopy looking things. So there is a bit of that going on. But if you look in the big picture, it's mainly serous. While we're here though, this is actually a good example of probably one of my favorite terms in anatomy is the description of, if you look at how this, so here's the mucus cells. The mucus cells are these gloopy looking things. The serous cells are these cells that make the amylase. They have that kind of granule. They stain this kind of dark granular. It's not uncommon for the serous cells to make these little half moons over the mucus cells. So we have the little serous cells making a half moon, and their official term, let me see if I can draw it. They're called serous. They're made out of serous cells. Serous. And half moon is demilune. I don't know, there's something poetic for me about that. The serous demilunes. I always kind of smile when I see them in the in the salivary gland tissue. So serous demilunes, the serous cells that make the amylase, making these little half moons on these gloopy mucus cells that are making the mucus part. So, like I said, let me clear this. In general, if we're looking at this gland, it's mainly serous. It's mainly these dark granular cells. If I go to the sublingual gland, it's going to be very different. If I go down and look inside here, you can see like, oh my God, way more serous. I mean, I'm sorry, way more of these mucus cells, these kind of gloopy looking cells, and not so many of those dark staining serous cells. Do y'all see that? You know, in fact, can I even find a demilune? Let me take a look. Yeah, okay. There's a little serous demilune sitting on top there. Oh, there's some more over here. But as you can see, preponderance of the cells in here are making mucus, you know, as well as we can see lots of ducts made out of simple cuboidal epithelium. These are more serous demilunes here. When we get into the submandibular gland, there I will find, I have prettier pictures of this. In general, the slides on this site are great, but I actually can say I have better slides of the submandibular gland, although you're not going to be able to zoom in, but I will show you some nice pictures of that I will upload as well onto the canvas. But if you look around, it's kind of a mix. There's serous cells, there's mucus cells, you know, so it's kind of a mix. And again, lots of ducts. And again, this one is not stained so well. So the ducts are actually, the ducts are kind of distorted. I don't like this slide that much. There's this here, you can see the duct. So glands. So those are under exocrine glands. You know, you should be able to recognize a salivary gland. I would feel okay putting a salivary gland on your final. You know, definitely, I would say even on your quiz, you're going to have to recognize at least what layer of the GI tract you're in. If there is a picture of, you know, of some part of the tube and there is a pointer, say it could say, what layer is this? What kind of tissue is this made out of? So you should definitely know the different layers, be able to figure out where are you, and then what kind of tissue makes up that layer. Is it the dense connective tissue of the submucosa, or the smooth muscle of the muscularis externa, or the epithelium and loose CT of the mucosa? So you should be able to kind of know your way around that.
I have a quick question about the mucosa.
Uh-huh.
Is that referring to the 3 layers? If you just say mucosa as an umbrella term, is that the epithelium, the lamina propria, and then the muscularis mucosa?
Yes, exactly.
Okay, thank you.
So yeah, and in general, there's not always a muscularis mucosa. There's almost the lamina propria is pretty much any kind of mucosa, because any epithelium is always sitting on top of a connective tissue. So there's always kind of a loose connective tissue right under it. Like that's what we call it even in the trachea, right below the pseudostratified columnar epithelium. We say like the lamina propria underneath it. So give me 2 seconds here. Boom. Anatomy, anatomy. Nervous. I was sorting through some of my stuff. And wait. Oh. Where is it? Oh, here it is. Okay. And it's here. Okay, so I am going to show you a couple of slides that are my own slides, which I'll upload for you, but let me share this with you now. Okay, need to find share, stop sharing that. And I'm going to share the screen. Okay, so a few things. One, so this is a picture of the submandibular gland, and it's just showing really nice. You can see a variety of the things we've been talking about. You know, you can see ducts, cuboidal epithelium making tubes. You can see blood vessels. This is going to be a vein because it's got really thin walls and it's full of blood cells. Here we can see mucus cells, these gloopy looking cells, along with the serous demilunes. So you should be able to like look at a picture like this and kind of orient yourself and know that you are in a salivary gland. You should know that the darker staining cells make the amylase, the enzyme. The lighter cells make the mucus. The things with cuboidal cells are ducts. Clear out. Oh, I did not get rid of my little. Yeah. So this is actually just, this is showing villi. You know, this is actually in the ileum, and you can just see the simple cuboidal epithelium coming up and down. This is going to be the lamina propria, kind of this loose connective tissue right under there. Submucosa is all of this stuff here, down into the muscularis down here. It's not stained as nice, but it still shows all the parts and pieces and parts. We'll do liver. This is liver. We'll do liver later. We'll do liver. This is typical looking colon. Here you can see these crypts going down, and these are all goblet cells. Basically, the crypts are lined with goblet cells. It's the main cell in their columnar epithelium, in order to just have lots of mucus going up to keep your, basically kind of greasing the skids for your poop to be making its way out. If you look underneath, again, there's going to be muscularis. Here's submucosa making these folds here. If you cross section it, so this what's really kind of fun, if you cross section this, instead of seeing these tubes from the side like this, you're going to look at each one. It actually looks like flowers. It's one of the prettier things. Where do I have it? Here. So this is it. So again, here is the colon. Here's showing the crypts going down. Here's what happens if you're cross-sectioning them, where you're looking at the gopher holes that have been cross-sectioned. And if we just zoom in on the cross-sectioned thing there, you get this, where it looks like a bunch of, these are like goblet cells. This is basically looking down the lumen of the crypt, looking down, it's like, you know, going down the gopher hole. These are the columnar and goblet cells. This is the loose CT of the lamina propria that's underneath the epithelium. And it looks, again, it looks like a field of flowers, and it's like there in your colon. So I always thought that's really cool. Right, so this is where all your poop is going through. It's actually going all over this. This is, yes, this is, if you're going to post it to Instagram, you might want to put it through a filter or something. So this is a cross section of a decalcified tooth. So this is the dentin. This is the pulp cavity. And if you start zooming in, it's kind of hard to see, but those tubules are actually this texture. The next slide actually is going to be looking at the dentin, but at a higher magnification. You can start to see now more of the dentinal tubules. So again, you have like these cells that are maintaining the tooth matrix, and they have little tubules that are running through the tooth, kind of like the osteons if you were in the bone, but you're in the dentin of the tooth. Dentin is a lot like bone. It's like the bone that makes up your tooth. I think I might have even one more, maybe not. But this gives you an idea. You know, a cross section of a tooth is actually a little more interesting than you might think. Let me see if I have other things in here that would be interesting. This one. So this is large intestine again. Here you can see the crypts with the goblet cells and the mucosa. Here you can see the submucosa, the dense connective tissue. Below that, the circular and longitudinal layers of the muscularis externa. And these things in between, these are definitely the myenteric plexus of Auerbach. So these are the neurons that are making up part of the enteric nervous system. And again, I always think it's pretty trippy that your nervous system is not just your brain. Your brain does a lot, and your spinal cord, they're important, obviously. But serotonin is actually the main neurotransmitter that is used by the enteric nervous system as well. Serotonergic things can actually mess up your digestive processes for reasons completely independent of messing with your brain, because it's actually can be affecting the neural networks that are just located in the walls of your intestines. But yeah, it's always, thinking about the different parts of your body that are kind of more independent than you think. Like, the leukocytes, the white blood cells are kind of doing their thing to keep you going. You've got all your little primitive, again, I like when they call it the primitive gut brain. Your intestines actually have a little bit of their own kind of know what to do without having to check in with the big brain upstairs and take care of business. So I think that, for our lab, I'm going to probably cut you loose now. What I think you should do is go and spend some time looking at these slides, see if you can start getting comfortable recognizing the layers, knowing what they are. Think about the basics of the digestive system, but know that we're going to be looking in detail at the stomach, the small, large intestine, liver on Thursday. So you don't have to think about all that detail. Right now, today was supposed to be about kind of setting the stage and also getting to know that, I think it's fun spending some time with teeth, because we rely on them a lot. And and I remember wondering how come we don't get extra teeth. It's a bit of a drag that we have those little enamel organs. Obviously, you can do it once. Your teeth can fall out and you can grow a new one. So it's not like it's not possible. You grow your first set of teeth. And again, other non-humans grow teeth all the time. It just happens to be the way we ended up. The little enamel organs kind of degenerate after you get your secondary dentition. So that's all she wrote. Once, if you lose a tooth, then you end up with a bridge or a implant or whatever. Another, I don't know if I can show, this is actually kind of cool. I don't know if this will show up. Enamel is this cool mineral, and this is a UV light. Actually, your teeth fluoresce really cool. They end up becoming this kind of eerie, glowing green. Like if you have, like I have a cap and a bridge in there, and you can see exactly where they are because the ceramic just looks like dark purple in the UV light, but your teeth are all like popping like crazy. So it's fun. If you have a UV light and you're just kind of bored, I would recommend look in the mirror and shine it in your mouth. It's kind of fun. Or at least I find it fun. So
I have a question about the teeth. What makes the teeth in your gums? Like, what's in your mandibular thing? Is it like a hair?
Say it again.
Is it like the hair roots? Like there's stuff underneath it?
No, so it's those periodontal ligaments. So there's little fibers going in between, going from the bone of your maxilla and mandible. So like here's the socket. Like let's say this is the bone, and your tooth is in there, and then there's little fibers that connect from the tooth to the bone, and it's basically a synarthrotic fibrous joint. So it's just the tooth and the bone are held together by very short little fibers running between the two, but so short that for the most part, the tooth doesn't move.
Okay, thank you.
Sure. So any other comments, questions, or otherwise?